everyone. It is my pleasure to introduce Dr. Gregory Dussor, who is an international leader in the study of migraine pathophysiology, using a variety of preclinical, behavioral, electrophysiological, and biochemical models with an overarching goal to identify therapeutic targets to alleviate migraine pain. Dr. Dussor is a Eugene McDermott Professor and Department Head of Neuroscience at the University of Texas at Dallas. Earned his PhD from the University of Texas Health Sciences Center in San Antonio, followed by postdoctoral training at the Oregon Health and Science University. Prior to arriving to UT Dallas School of Behavioral and Brain Sciences in 2014, he held faculty position at the University of Arizona College of Medicine. Received numerous awards, including Future Leaders in Pain Research Award from the American Pain Society, serves on editorial boards of leading journals, including Pain Reports, and the Journal of Neuroscience, to name a few. Additionally, is an advisory board member of the Migraine Research Foundation and the Association of Migraine Disorders, Migraine Science Collaborative. And it is my great pleasure to welcome Greg here.
Thank you. Apologies for that mouthful. And thanks for the invite, and thanks to all of you here in the room and online for listening. So I'm going to talk about migraine today, as you heard, topic that may be personally interesting to you. I'll talk about some statistics to get going. I usually like to start presentations with information from this study here, which is the Global Burden of Disease study from the World Health Organization. And so what they do is every few decades, they rank prevalence and burden caused by diseases and injuries worldwide. And so this is 328 diseases across the earth in 195 countries. These are the top 6 most common diseases on earth. If number 1 doesn't, if you don't recognize number 1, that is actually cavities of permanent teeth. So that's the most common disease on earth. Migraine is number 6. Just because something's common doesn't mean it causes disability. So here's the ranking of the top 6 most disabling diseases on earth. Again, World Health Organization numbers. Low back pain number 1 and migraine number 2. So what we're going to talk about today is the 6th most common and 2nd most disabling disease on the planet. So here is the timeline of what a migraine attack looks like. There are different phases to an attack. There's a prodrome phase where people will have any number of symptoms that can alert them that the migraine is coming within some number of hours or maybe a day. Yawning, fatigue, food cravings, a variety of things. There is, of course, the headache phase, which causes people to draw images like you see on the bottom left. There is, of course, the unilateral throbbing headache, very intense, 8, 9, 10 out of 10. There's also sensitivity to light and sound. There's nausea and vomiting. There's cutaneous hypersensitivity. People don't want to touch their skin. They don't want to brush their hair. They don't want to touch their face. This is 4 to 72 hours. Many people have migraine attacks that go much longer than this, so this is just in the diagnostic criteria. And then there's a postdrome phase where people often report feeling hungover. There's weakness, there's tiredness. It takes them some amount of time to get back to their normal cognitive function. So you add all of this up, and this may be many days of a week. If people are lucky, they're lucky they don't have migraine at all, of course, but if they're lucky, they only have 1 or 2 of these a year. But there's a specific subset of the condition known as chronic migraine, where it's 15 or more migraine days per month. That's a disabling condition. Even if you get a couple a month, it's a disabling condition. That's why it's number 2 on the list. One of the interesting things about migraine is that attacks can actually be triggered by specific events. And here's a representative bar graph. There's many, many studies that have looked at this. And so if you look at the percentage of people who report different kinds of triggering events, the most commonly reported trigger in almost all of these studies is stress. Some relationship between stress and migraine attacks that we don't fully understand. I'll talk about that in a bit from the animal model perspective. Changes in hormones, big problem. Not eating, so skipping a meal, timing when you eat inappropriately. Weather, changes in sleep. So people sort of recognize that if you don't get enough sleep, this might be a migraine triggering event, but actually too much sleep is also a migraine triggering event. So they have to titrate the amount of sleep to a certain number of hours. All kinds of other things, scents, lights, alcohol, heat, food, exercise, seemingly unrelated things. So it's kind of hard to make a hypothesis about how this is actually happening, because many of these are unrelated to each other. But these are all very common triggering events for migraine. What's interesting about this is that everyone experiences these triggers. Not everyone gets migraine. So what is it about the migraine nervous system that allows an attack to happen in response to these normal events that we all experience? That is a big part of what we don't understand. Migraine is more common in women. So this is a graph of age on the x-axis and then prevalence on the y-axis. And in red is males and in blue is females. And so early in life, males and females are relatively similar as they are later in life. But between the ages of certainly 15 to about 60, women have much higher prevalence of migraine than men. So this is typically thought of as the ages of women when they're normally cycling. Right? So again, a link to hormones. This is a very widely reported statistic in the field. Here's another one. You look across the monthly cycle, you can see a graph of changes in hormone levels, estrogen and progesterone. And then the shaded area is when women are most likely to have migraine, which is in the late luteal phase. Interestingly, actually not when estrogen and progesterone are high, but when they're falling or low. So that's confusing. Here is a graph of not just the menstrual cycle, again, hormone levels in the lines there, lines going up and down, of course, shaded areas of when women are most likely to get migraines. And so on the left, you can again see the monthly cycle, but also during pregnancy, many women who have migraine before they get pregnant, they often get better during the first couple of trimesters. In the late phase of pregnancy and certainly after birth, many times this will come back, maybe come back even worse. And then around menopause, the frequency goes up, but it can stabilize after menopause. So this is a lifetime disease. It's not a disease of aging. This is something that kids get. And in fact, this is a disease that may actually get better as people age. And the reasons for that are not yet clear. So let's talk about how migraines are actually treated. So if we were in ancient Egypt, 2500 BC, and you had migraine, the way that you would be treated would be, there would be a clay alligator. Herbs would be put in the alligator's mouth. This would be strapped to your head, and this would somehow be therapeutic for migraine. And there's evidence of this in the Evers Papyrus, the oldest known medical text. It's kind of hard to see here. You can, if you look at it closely, you can see sort of demons in the head of the person with migraine, and there's representation of these alligators. And this is actually an alligator that was found at an archaeological site. One of the examples of what would be used to treat migraine. Now, we've of course come a long way. We're 2025 AD now, and how are we treating migraine? We have things like the MigraCap, which is the gel pack that you put in the refrigerator or the freezer or the microwave, and you heat it and cool it, and you strap it to your head. So there are still therapeutic approaches for migraine that are actually not that different than what was done in ancient Egypt. To be fair, there are actually drugs that are used for migraine. I don't pretend that the people in the room are going to be able to see all of the words on this slide. I think the point, though, is that there are a lot of different categories of drugs that are thrown at this. On the left-hand side are drugs that are used for the acute treatment of migraine. So if you only get a migraine a month, 2 migraines a month, you carry around drugs with you. When you think you're having a migraine, you take those drugs. These are anti-inflammatory drugs. These are triptans. This is a category of drug that is specifically used for migraine. It's not used for anything else. They're serotonin 5-HT1B and 1D agonists. There are ergot derivatives. That's an interesting story that I can talk about if anybody's interested later. There are new CGRP-based drugs, and these have exploded in the last few years. These are used acutely. There's devices. On the right-hand side, if you get more than, say, 7 or 8 migraines a month, you may take a drug every day to prevent the occurrence of the attack in the first place. And so with the exception of CGRP-based drugs, which are on this list, none of these drugs were designed for migraine. So these are anti-epileptic drugs. These are antidepressant drugs. These are blood pressure lowering drugs. No theme between them. We don't really know why it is they work for migraine. Poorly understood thing. So as I mentioned, CGRP, this is calcitonin gene-related peptide. This is now a very clear target of migraine drugs. And there are 8 just since 2018, there are 8 new FDA-approved drugs for migraine. They are all targeting this peptide. They either bind to the peptide itself or they block the receptor for the peptide. If you have the TV on ever, you can't escape commercials for these drugs because they're just every single commercial break that comes on, you will see either a singer, you'll see a talk show host, you'll see, I don't know why she's famous. That's another story. You can't escape. They're amazingly common now, and they're actually helping a lot of people. But there's still, we already know, large numbers of the migraine population that either don't get any relief at all from these drugs, because presumably CGRP is not the problem in that fraction of people, or the relief that they get is not complete. So we still need new therapeutics with mechanistically distinct targets for people with migraine. So where does migraine actually come from? What is the substrate of migraine? And we can focus here at least on the CNS. This is a diagram from a review in 2022. I again don't think that you're going to be able to see all of this, but often when you see a diagram like this with arrows everywhere, it essentially means you don't understand what the pathology of the disease is, right? And I think that's probably a fair summary of looking at this head diagram. We don't actually know where the disease originates in the nervous system. And I would argue it's really not even fair to only have the head on here, because there are sensory abnormalities throughout the body that come along with migraine attacks. There's gastrointestinal problems that come along with migraine attacks. The origin of migraine for many people is actually in the GI tract. There are many food triggers. So this is a body-wide disease that we really just don't understand. So as complicated as this is, what we try to focus on in my group is at least a part of the disease, which is the pain phase of the disease. Migraine is not just headache. There are, of course, a lot of other things that happen, we talked about a minute ago. But we're interested in the pain phase because this is the reason why people aren't going to work. This is the reason why people lock themselves in a quiet, dark room and wait out the attack to be over. The pain phase is really the big problem. And I think that if you talk to people with migraine, they will tell you the other parts of the attack are problematic. If I could get rid of the pain phase, I could at least function. So what do we know about where the pain of migraine, or really any headache, actually comes from? This is a very famous publication in the field, the headache field. It was published in the Archives of Surgery, which is now JAMA. And it was published in 1940. And it was a couple of neurologists that what they did was across a number of humans that were having open skull surgery who were conscious. What they did was probe at various structures inside of the skull and ask the patients, does this hurt? And so this is this is an amazing paper. Anybody who wants to see it, I can send it to you. It's unbelievable. This is just figure 5. There's a lot of amazing figures in this, but if you can see this here, the dark circles are areas that were painful. And it took this group a number of patients to be able to get the entire inside of the skull, because it just depended on where any given person had the skull open. The black circles are where they would stimulate that was painful, and the open circles were where they stimulated that wasn't painful. And the conclusion from all of this work was that the meninges that cover the brain are the pain-sensitive structure inside of the skull, and essentially nothing else inside of the skull is painful. The other amazing thing that they did was they asked these patients, when I probe wherever they were probing, where does it actually hurt? And the shaded area in the head drawings around the edge are the location of the referred pain following stimulation of the meninges. And it turns out that the most commonly reported location of the pain of migraine is around the eye, in the temple, somewhere in this part of the head. And interestingly, you can recreate pain in those locations simply by stimulating the meninges. Right, so this is really where, for decades now, we've thought the pain of the migraine attack actually comes from, is activation of sensory neurons that innervate the meninges. The question is that what is actually activating those neurons? That's something we don't know. Now, I will say this study was replicated in 2018. I think these authors thought, we're smarter now, we have better technology, we can do this better, and maybe we'll find something different. Essentially, they found the same thing. So decades later, this has actually been replicated. We still think that the primary pain-sensitive structure inside of the skull is the meninges. So how do we actually turn this into a preclinical model? It turns out that we can actually stimulate the meninges of mice. I'm not going to get into the details of exactly how we do this. We can apply really any kind of stimulus that we happen to be interested in to the meninges of a mouse, and then measure a behavioral output. And then the question is, what is the behavioral output? How do you know that a mouse is having a headache, let alone a migraine? The endpoint that we use commonly, we have a couple of different endpoints. The primary one that I'm going to talk about today is periorbital skin hypersensitivity. So I told you a few minutes ago that in humans, during the headache phase of a migraine, they don't want to touch their face, they don't want to touch their scalp, they don't want to brush their hair. The skin of the head is incredibly sensitive. Turns out that is something we can measure. Obviously, we can't talk to a mouse and say, are you having a headache? We have to extrapolate from their behavior. Skin sensitivity in the head is something we can measure because we have calibrated filaments that we can probe the forehead with and measure a threshold of a response. And if that threshold goes down, it means the head is hypersensitive. This is consistent with headache, of course. Obviously, there's going to be limitations of what you can extrapolate from an animal's behavior. But this is the kind of experiment that we would do. So here are the kinds of graphs that I'm going to show you from the animal models. And males are on the left, females are on the right. We do a lot of work comparing the sexes, given the sex differences in the prevalence that I showed you earlier. What we used here when we first started sort of optimizing and developing this model was a cytokine called interleukin-6. I'm not really going to talk about that cytokine today. That's a cytokine that you probably are familiar with because it's implicated in just about every disease on Earth, migraine included. But we used it as a stimulus to apply to the meninges. And if you look at the graph here, the y-axis is facial withdrawal threshold. So this is the response threshold to these filaments that we can probe the skin with. And a drop down the y-axis indicates hypersensitivity. So if the data go in the negative direction, that means the animal became hypersensitive from whatever it is that we did. So we apply IL-6 to the meninges of the male and the female mouse. You can see the red line there is the IL-6 group. We see a drop in the facial withdrawal threshold for many hours, a couple of days actually. That recovers, and then we can come back and stimulate the meninges again now with something that is typically sub-threshold. But now the animals are actually sensitized to a stimulus that they wouldn't otherwise respond to. So the black is a vehicle application to the meninges, doesn't cause any kind of hypersensitivity response. And when you come back with that second stimulus, in this case, it's a pH7 stimulus, the details of that don't matter. If you just apply a vehicle to the meninges, when you give a pH7 stimulus later, nothing happens. Right? So we've sensitized, caused a behavioral response here, and we've caused a later sensitization to another stimulus. This is one of the major types of preclinical models that we use in this field, us and others. The other model is based on the graph that I showed you earlier, taking the observation that stress is the most commonly reported trigger of migraine, and basically asking, can we backwards translate this into animals and use this as a stimulus to cause a migraine-like phenotype? So what we do is we repetitively stress mice. It turns out this is pretty straightforward to do. There are many different ways of inducing stress that people have used over the years. And so what we do is we habituate the mice to the experimenter, to the environment, to the chambers for a couple of days. And then we do 3 days of stress, 2 hours each day. So it's 3 consecutive days. And what we do is it's a restraint protocol. We put the animals in a tube and confine them to the tube. This is considered a moderate stress in the animal literature. There's much more extreme stressors that you can use, and there are much milder stressors. Humans sort of look at being in a tube and restrained as a much more stressful thing, but we're not burrowing rodents. Rodents actually like being in small environments. They just don't like being trapped there. So the confinement is the stress part. So this is what we do, and then we test the animals over days following the stress protocol. And then what we also incorporated into this model was a second stimulus, like I showed you a minute ago. But in this case, the second stimulus is injection of a nitric oxide donor. The reason that we do this is that nitric oxide donors are very well known in humans with migraine to be reliable triggers of migraine attacks. So classic example of a nitric oxide donor is nitroglycerin. This is a drug that's classically used for chest pain. People with migraine can't take these drugs because 75% of migraine patients will develop a migraine attack within 5 or 6 hours of being administered a nitric oxide donor. So contraindicated in people with migraine, but it's a research tool. And if you were to want to, for example, have a human in a scanner while they're having a migraine, how do you know they're going to have a migraine while they're in the scanner? You can actually induce a migraine in a research subject by giving them nitroglycerin or any nitric oxide donor, and you time that to when they're in the scanner. It's about 5 or 6 hours delay, and this has been used many times in the literature. So what we're trying to do here is to create a model that reflects stress being an important trigger of migraine and also incorporates a pharmacological trigger of migraine. So the model sort of looks like this. The pain behavior here is the y-axis. We baseline the animals. These are sort of sometimes referred to as multi-hit models. The first hit in our case is stress, repeated stress exposure. That causes a priming event somewhere in the animal, in the nervous system. There's a sensitized phase, and then you can come back to the animal later with another stimulus that it wouldn't otherwise respond to, but now it's sensitized or primed to that second stimulus. This is the first paper that we published using this particular model. And here are what the data look like. Again, males on the left, females on the right. The facial withdrawal threshold is again on the y-axis, and a drop in that threshold indicates hypersensitivity. So the first time point on the x-axis is the baseline. 24 hours, which is the first time point on the x-axis, is 24 hours after the 3-day stress protocol. So we do the 3 days of stress. You can see that drops the withdrawal threshold. It sensitizes the animals for many days. We wait out to about 2 weeks, and then the SNP is a nitric oxide donor. We give those animals a nitric oxide donor, and only the animals that were previously stressed will develop a response to the NO donor. Animals that have not been stressed show no effect at all to that dose of the NO donor that we used. And so you can see the reestablishment of the hypersensitivity in both the males and the females, but only in the animals that were stressed. What we've been trying to do with this model is to try to optimize and further validate it by using drugs that we know work in people. And so one of the things that we did in this original paper was we treated the animals before we gave the nitric oxide donor with a CGRP monoclonal antibody. And we did this in collaboration with one of the companies that makes these antibodies that are approved for use in humans. So they gave us the antibody. We dosed the animals before we gave the NO donor. And the black line there on either graph is the efficacy of the CGRP antibody against the sensitized response to the NO donor. And on the left, you can see an effect in the males, not really that impressive, but it's there. Much bigger effect in the females. So this was interesting to us. It still is interesting to us. I'll come back to this in a second because there's something interesting about CGRP that seems to be working differently in males and females. Yep.
Specific to, how do you know this is specific to this pain protocol? So if you had administered pain in another location, you can see an effect.
Pain being the
So an adverse stimulus to, like, you know, a heel pain or something, or of course they're kind of all that. But I guess I'm just wondering to what extent is stimulation of the head versus.
Yeah, I mean, so I guess the question maybe people can hear is, to what extent is this specific stimulation of the head? So I'm not actually showing you in these graphs hind paw hypersensitivity. We stopped doing this. We did actually do this for a while. We tested the hind paws. They also get hypersensitive. There's nothing really head specific about stress. There shouldn't be anything head specific about stress. And in fact, in people who are having migraines, the sensitivity is not just limited to the head. Their sensitivity spreads throughout their body in many people. So it's a good question though. On the dural stimulation model, that is a more head specific. So if we did that same injection in the paw, we see paw hypersensitivity, but we don't see hypersensitivity in the head. So, you know, there is still some questions that can be asked about what is the mechanism. At least, certainly in the case of stress, stress is a body-wide exposure. It should impact the entire body. We're sort of framing this in a headache context because of the connection to the human disease. But there's nothing specific about the impact of stress on migraine. Stress impacts all kinds of things, right? So this is almost certainly a messy model. I think people with migraine are also mechanistically messy. Right? So, I think there's some value to that, and I can say that pharma has thought that there's some value to this model because they were very interested in it. But yeah, there's still a lot of questions that can be asked about how specific this is. Okay, so we've also been trying to further validate this with other drugs that we know are used in humans, and so this is just the same kind of graph. Might look a little different because a different person did it. The blue is the control group, and the black is sumatriptan. This is a member of the triptan group of migraine-specific therapeutics, and so we get some attenuation. It's certainly not complete. Some attenuation of the behavioral response with a migraine-specific drug. This is a CGRP receptor antagonist, and here we're actually giving them earlier in the time course. I should point that out. CGRP receptor antagonist works in this model, as does a non-steroidal anti-inflammatory drug, ibuprofen. So we are able to show that in these models, the drugs that we know work for migraine in humans also work in the model. Right? This is part of what we try to do for validation of these models. It's not perfect, of course, but it's one of the better ways that we can look at a model and say that it has some validity to it. So in the dural stimulation model, one of the things that we did early on was ask questions about CGRP. So yeah, Sean.
Question. In the previous models, because there are different types of drugs that you're testing, to what extent the recovery after the introduction is dependent on the the change in baseline?
Yeah.
Ibuprofen, we think, wow, that's a massive decrease in baseline before it disappears. So, and those kinds of that possible signature to look at?
Yeah, it's a good question. The sensitivity of this model is not enormous because there's a finite number of filaments that exist. They're not a continuous range of filaments. And so, the difference between one value and the next value is a finite response, right? That influences the statistics that we are able to run on these kinds of data. So, sometimes you see an effect that might look bigger than what you would predict. Is that a matter of sensitivity of the model? I don't try to make that much about the magnitude of the effect that we see. One interesting question is whether or not the full establishment of the first phase is necessary for the full establishment of the second phase. That I can tell you from other types of work that we've done in a non-headache context, that doesn't appear to be true. Because we can pharmacologically distinguish the two phases. So I don't want to say that the first phase has no role in the second phase. It certainly does, but it's not the only reason that the second phase is established. So we're interested in sex differences. We're interested in the mechanism of CGRP. As I told you, that this is a very widely used target of drugs now. But I honestly can't tell you that we know how CGRP contributes to migraine, where CGRP contributes to migraine, when it contributes. We just don't know. It's remarkable that there are so many drugs now that work, but I honestly can't, and I don't think anyone in the field, it's not just me, I don't think anyone in the field can tell you exactly how and where and why CGRP does what CGRP does. So we asked a relatively straightforward question, which we thought was, what happens if you just put CGRP on the meninges and using the dural stimulation model that I told you earlier? And what we saw, the red there is the female response, and the blue is the male response. So same CGRP, same dose, same experimenter, same lab, same room, everything, just different sex of the animal. And there's a remarkable difference in the way that the females and the males respond to just this very straightforward single stimulus exposure to CGRP. I'm not going to tell you that I understand yet exactly how this works. We've been working on this for a while and we still haven't figured it out. So I'd love to be able to tell you, but I can't. However, what I do think is interesting is that there are now starting to be papers showing that there might be differences in the efficacy of these CGRP-based drugs in humans, males and females. And so we published our paper in 2019. These are two different papers from 2023 and 2024. And what the authors are arguing is, that these drugs might actually work better in women, And B, we may not actually have enough evidence that they work in men to be prescribing them to men. Maybe that's a little bit of an extreme statement to make. But what the authors of these papers did was they used the data from the drug companies that were submitted to the FDA as part of the FDA approval process, right? That these data become public later on, and they're available for review. And what the authors did was they looked at 3 different CGRP receptor blockers. And the graphs here are pain freedom at 2 hours. So there's women in red on the left, and then there's men in the sort of the middle in blue. And skewing to the right favors the drug over the placebo. So what you can see is that in females, in women, there's pretty clear skewing to the right favoring the drug, and in men, not so clear. And when you graph these together as a line graph, there's separation between those two, and as a bar graph, you see a bigger effect. So I can't tell you that I know what the mechanism for all of this is, but it's at least nice to know that the work that we're doing in the animals actually has some relevance to the human disease and maybe to the therapeutic opportunities that we have in the human disease. Otherwise, we're just working on mice, and this has no translational potential. So, so this is still a work in progress, and we're still looking at this. One of the other things that we've been looking at in terms of sex differences in these models is the hormone prolactin. So this is a letter written to the Lancet in 1973 by Dr. D.F. Horobin in Newcastle upon Tyne. And it's been a while since he did this, but basically what he said was, he noted that prolactin levels are highest in the early morning. They're elevated by stress of almost any kind. And I wish to suggest that the events of a migraine attack, while not actually caused by prolactin, can occur only in the presence of high or changing prolactin levels. And that a trial of, this is a drug called bromocriptine, it's a prolactin lowering agent, seems to have remarkable specific effect in reducing prolactin, that this should be tested in people with migraine. It took us 52 years or so to do some experiments on this. I had a colleague who was working on prolactin in another type of project, a non-headache project, and we got together and started collaborating on this. And one of the first things that we did was apply prolactin to the meninges of the mouse. And similar to CGRP, on the left is the response in the female. The black is prolactin, the red is vehicle, and on the right is the male. Remarkable difference between females and males, again, as we saw with CGRP. I'm not going to go into the rest of what we did in this paper, but there's actually some interaction between CGRP and prolactin in the meninges. That was the focus of the paper. So there's something about these two substances that are working very differently in females than they are in males. We've done more work with prolactin, and I think are maybe in a better position to be able to talk about mechanistically why this might be occurring. And so one of the tools that we have for the prolactin work is we have a mouse where there is a genetically encoded red fluorescent tag in cells that express the prolactin receptor. So what you can do is you can look at tissue from the mouse, and anywhere that you see red fluorescent tag, red fluorescence, that those cells express the prolactin receptor. So these are images from the meninges of a mouse. Part of the meninges of a mouse is the dura, the outer layer of the meninges. And it may be hard to see in this room, but on the left is a 10-week-old female dura, and on the right part is a 10-week-old male. And there's a lot of red here. There's a lot of sort of dots that are spread around. Those are non-neuronal cells, but the filaments that you can see on the left are nerve fibers innervating the meninges that express the prolactin receptor in the female, and they're remarkably absent in the male. So this suggests that there's differential expression of the prolactin receptor on these nerve fibers in the meninges between females and males. So we can actually take out the cell bodies of those neurons that project to the meninges. These are in the trigeminal ganglia. We can culture these in a dish, and we can do electrophysiology on them. We can stimulate them with an electrical stimulus. We can measure action potential firing, and we can incubate the cells with prolactin, and then we see more action potential firing, but only in female neurons. So the bar graph on the bottom right is the normalized data. And if we use a vehicle or prolactin in the male, we see the same number of action potentials being fired. But in the female, if we treat female neurons with prolactin, we see an increased number of action potentials. We've increased the excitability of those neurons simply by incubating them for a couple of minutes with prolactin. And this is consistent with differential expression of the receptor. So it's possible that the behavioral effect that I showed you a minute ago is mediated by differentially expressed receptors for prolactin in the meninges of females and males. We've also tested this in the stress model that I talked about. And so Dr. Horobin, 52 years ago, noted that stress elevates prolactin levels in the blood. That's actually one of the best ways to elevate prolactin levels outside of pregnancy and nursing. Stress is one of the best ways to elevate prolactin levels in the blood. And so here are data of serum prolactin levels for mice. The 4 bars on the left are females and the 4 bars on the right are males. And under control conditions, just normal conditions in the mouse, the prolactin levels in the female serum are much higher. The y-axis is actually broken twice. Because in stressed females, the prolactin levels in the serum go up dramatically. In the male, there's hardly any prolactin circulating in the male, but it does go up in the serum of males after stress, right? Just not anywhere near the level that it does in females. If we treat with bromocriptine, which is the prolactin lowering agent, this is a D2 dopaminergic agonist, and it activates dopamine receptors in the pituitary circuit that controls prolactin release. If we treat with bromocriptine, we have a significant drop in the prolactin levels in the female serum. We also have a drop in the amount that prolactin goes to after stress, right? So bromocriptine is just lowering the prolactin levels overall. Also does the same thing in males, it's just working in a different concentration region. So we're basically trying to do the experiment that was proposed 52 years ago in the mouse and say, does treating with bromocriptine to lower serum prolactin levels have efficacy in this particular model? So here's the female. These are wild-type mice. There's nothing unusual about the mice. This is the same stress protocol, and the blue lines at the bottom are the control, the vehicle, treated animals just exposed to stress. The pink, should be pink, open circles are bromocriptine treated females. So you can see an attenuation of the initial response to stress, and then almost a complete block of the response to the NO donor in the second phase of the model. If we do the exact same experiment in the male, hardly anything happens, right? So this is the same mice, same stress, same room, same everything, same experiment, or same drug. It just doesn't really work that well in males. So is there any translational relevance to this in humans, which, this is a question that we're always trying to ask in these models. There have been a number of studies over the years that have looked at a connection between prolactin and headache disorders. This is all circumstantial evidence. People get tumors of the pituitary that cause excessive amounts of prolactin to be secreted, causes a hyperprolactinemia. This is the reason why these prolactin lowering drugs were developed in the first place, is to try to suppress that prolactin release from the pituitary. So there's a number of papers that have been shown that people with these tumors have headache or worsening of migraine as one of their prominent symptoms, and that when you treat them with the dopaminergic agonist, their headaches get better. Okay. My colleagues and I, we've written 2 systematic reviews now of all the preclinical studies and all the clinical studies that have made a link between these two. We're trying to make the argument that this actually should be tested, again, 52 plus years later. We were sort of planning to get into the stages of doing this, but here's a paper. This was first presented at the European Headache Congress in December. I was there. This paper is now on medRxiv. It's not yet published, but they did a prospective randomized controlled trial, 33 participants, 30 of them were female, of a prolactin lowering agent. In this case, they used cabergoline, which is a very similar drug that we're talking about. And they actually divided the group into episodic migraine subjects, which is less than 15 migraine days per month, or chronic migraine, which is more than 15 migraine days per month. And they see a significant lowering of monthly migraine days in the episodic group, as well as an increase in the 50% reduction in migraine days in the episodic migraine group, not in the chronic migraine. So why that is, I don't know. Maybe they would have gotten a bigger effect if they hadn't had 3 males in the study. I don't know that either. They sort of scooped us on trying. It would have taken us years to get this off the ground anyway, and ideally a clinical trial core facility on our campus, which we're trying to establish. But it's great to see that actually this ideas from the 70s turn into animal models, which I know they used as the rationale for their study because they cite us. it's great to see this actually turn into efficacy in humans. And so, I think there's a lot of potential around this. Drugs are available. People with migraine are available, right? This is something that can easily be expanded on, and I hope that actually ends up happening in the future. So now I'm going to transition into how we're actually trying to incorporate more human work and human tissue into the work that we do. And it's really originally based on this observation that has been around in the field for a while, that again, what is happening in the meninges to activate those sensory neurons to cause a headache? We don't know the answer to that question. One of the things that's been proposed is sterile inflammation. Sterile meaning it's not caused by bacteria, it's not caused by a virus, it's something endogenous that's causing an inflammatory reaction. That this is happening in the meninges in response to something. And part of it is immune cell mediated, and there's plenty of immune cells that are present in the meninges. Mast cells, a major type of immune cell, are one of them. And this has been a focus of people in the migraine field for years. And so there's been this idea that mast cell degranulation, so mast cells are interesting because they degranulate, they release their contents. That can act on all kinds of pathways and cell types downstream. But mast cell degranulation as part of the sterile inflammatory process is actually part of the contributing process to initiating the activity in these nociceptors innervating the meninges, and this is where the headache comes from. Not, of course, where the whole migraine attack comes from. That's in the CNS somewhere, but the headache phase, how do we activate those meningeal nociceptors? So this idea has been around for a while, many publications and many reviews on it. What we're specifically interested in is one of the potential downstream effectors of mast cell degranulation, which are protease activated receptors. So one of the things that mast cells release is tryptase. It's a protease. Mast cells release all kinds of things, histamine, prostaglandins, long list of things. But one of the major things they release is tryptase. And tryptase can act on a very interesting family of receptors called protease activated receptors, conveniently named because they're activated by a protease. But the receptor is a G protein coupled receptor. The N terminus of the receptor is extracellular. Yes. And the N terminus is cleaved by a protease. And after the N terminus is cleaved, part of it floats away, but the remainder of the N terminus now becomes the ligand for itself. So it's a very unusual G protein coupled receptor because it activates itself, but it only activates itself after it's cleaved by a protease. And once it's cleaved by the protease, it's permanently on because you can't uncleave it to turn it off. You have to pull it off the membrane. And so depending on where you cut it, it engages a different downstream signaling pathway in the receptor. So it's a super interesting family of receptor. Lots of different proteases can cleave these receptors. There's 4 different versions of this receptor. We happen to be interested in number 2, PAR2, because this is the protease activated receptor that is cleaved by tryptase. It also happens to be expressed in peripheral sensory neurons, which is relevant for pain and migraine. So we published a couple of papers on this. Other people have been working on this as well. It's not just us. I'm not going to go through all the animal data here. You can imagine that there are similar types of experiments to what I've shown you already. We can apply a mast cell degranulator to the meninges, and we can get a behavioral effect that we can block with a PAR2 antagonist or in a PAR2 knockout mouse. There's some role for PAR2 downstream of mast cell degranulation. We've established that. So what we're trying to do now is to turn this into a therapeutic opportunity. So I should disclose that Ted Price, my longtime colleague, and I are co-founders of a company that are developing antagonists to this receptor for pain, for migraine. So I clearly have somewhat of a bias here towards this particular pathway and this receptor. But I will also say that AstraZeneca, a major pharmaceutical company, just recently did a phase 1 clinical trial with a PAR2 monoclonal antibody for migraine. It was safe and well tolerated. They're now enrolling people in a phase 2 trial to test the PAR2 antibody for migraine. So it's not just us. Clearly, I have a bias. AstraZeneca is interested in this. Others are interested in this. I think this is a potential target, and they do as well. Otherwise, they wouldn't put a lot of money into a phase 1, now a phase 2 trial. We're actually hoping that their trial is successful because they will effectively validate this target in humans, which makes life much easier for those of us who are trying to follow this. So it turns out that when you look at sensory neurons in the mice, the receptor is actually only expressed in about 3% of sensory neurons. So this is an image, it's a section of sensory ganglia from the mouse. The top, the green is CGRP. ALK is the gene name for CGRP. So that's cells that express CGRP. The next one down is a P2X3 ion channel in red, another pain-related channel. And then the third row is F2RL1, which is the gene name for PAR2. You don't really see much label for PAR2. And the conclusions at the top of the slide is less than 3%. So if you make a pie chart of cells that express PAR2, really not very widely expressed. And this is part of the reason that many people in the field have dismissed the receptor, because it's just, I mean, the expression level is just not really high. You can take sensory ganglia out of the animal, you can culture them, you can look at how many cells respond to a PAR2 agonist. So 2AT here, sort of buried in here, is a PAR2 agonist. So how many cells respond to this PAR2 agonist? It's consistent with the expression pattern, really only about 3, 4, 5-ish% of the cells. So, you know, that's not very impressive. However, we're now, Ted and his group have really established this amazing tissue pipeline network with the Southwest Transplant Alliance, which is an organ donor facility here in Dallas. People are on life support, their organs are harvested, and then other tissues become available for research, obviously with proper protocols in place. So we're getting sensory ganglia from humans into the lab several days a week now, and we're able to do experiments with them, histological experiments, we do electrophysiology with them. And so what we've gotten a lot of so far are dorsal root ganglia, which are essentially on either side of each spinal level going down the body. Not the most relevant ganglia for migraine, because what we want are the trigeminal ganglia, which are at the base of the brain, but it turns out those are actually trickier to get sourced for reasons that I can talk about later. Maybe they're obvious to you, but tissue coming from the head is a little tricky. However, we think that the trigeminal ganglia and dorsal root ganglia are very similar. Every study that's ever looked at both has shown that they're similar. We do intend to do all of this work on the trigeminal ganglia and getting closer to being able to get them. But what I'm showing you now is expression pattern in human dorsal root. And the histologies here at the top, the pie charts down here are 4 different donors. And what you can see is it's somewhat variable, but at least 25% of the neurons in human express PAR2. So I've sort of been making the argument that we can use mice as a preclinical model to understand human disease. Now I'm telling you, mice only express this receptor in 3% of the neurons, and humans it's at least 25%, potentially more. You know, that's an interesting place to be. We can culture these human neurons. Again, we can apply the PAR2 agonist. That's here, 2AT. You see responses in about 40% of sensory neurons, again, consistent with the expression. We can do electrophysiology in human neurons. This is cultured primary human sensory ganglia in the lab. Electrophysiology from these, we can stimulate them, we can incubate them with the PAR2 agonist. You see increased electrical activity in these neurons. You see increased spontaneous activity in these neurons, and it's about 65 or so %. There's probably some enrichment process that's happening in the culture dish here to select maybe for cells that are expressing this receptor. But at the very least, this is vastly different than what we see in the mouse at 3, 4, 5%. These numbers are much higher, and likely we've been underestimating the role of this receptor probably in many sensory disorders because of its limited expression pattern. So what we're working with now, and so how do we rectify this, and how do we create a mouse tool that might actually help us to replicate better human disease biology? What we have is a mouse where the mouse PAR2 receptor has been deleted from the genome and replaced with the human PAR2, and it's behind the human PAR2 promoter. So what we see in these animals, this bar graph is just the expression in mouse and human. I've already shown you it's higher in human. But here is a mouse PAR2 expressed in mouse ganglia compared to the transgenic mouse where human PAR2 is expressed, and it's in a much higher percentage of cells. So just simply by replacing the mouse with the human version behind the human promoter, what it looks like is that we're getting an expression pattern that's more human in this. We're not arguing at this point that PAR2 might be a relevant target for migraine. That I think we've established, and other companies have established as well. What we're looking for now are tools that we can use to test PAR2 directed therapeutic whether they're antagonists or antibodies. And being able to do this in the rodent actually speeds things up dramatically. And so what we need is a tool where we can test this. So I think this is possibly one way. Okay, so I've thrown a lot of stuff at you. I'm going to stop because I want to have time for questions and discussion. And so just to recap, migraine is highly prevalent and leading cause of disability. The disease is very, very poorly understood. Obviously, we need to understand the disease better so that we come up with mechanistically distinct targets. I mentioned a few of them here. Preclinical modeling, I think, has identified ways that we can do this, potential novel opportunities for future therapeutics. There are some areas where these models are useful. I think it's also helpful to know where they're not useful. That's always true in animal modeling. And the last point I will mention, despite the numbers that I showed you on how prevalent this is, 16% of the Earth has migraine, and it's the second leading cause of disability. You would actually be shocked if you looked around at how many people actually study this as a disease. It's dramatically understudied across the Earth, right? And if I looked just in the animal world, if we go throughout all of the websites of all the research labs in the country, we look at how many of them actually state on their website that they focus on migraine, it's less than 10. Less than 10 laboratories in the country are actually studying this disease. And that's ridiculously inconsistent with the human prevalence in the human burden. So I always sort of make the pitch, at the end of talks now, that think about migraine in whatever it is that you do, whatever it is that you're interested in. If you have a cohort of 100 people in any subject, any study that you do, statistically, 15 of them have migraine, just numbers. It's all women, and it's a higher percentage than. So think about maybe how that might influence or be influenced by what it is that you're studying. I'm trying to pitch more people to work on this particular business. This is a relatively recent group photo of the lab. Of course, they're the experts. I just beg for money to pay for them to do what it is they do. I mean, they're the real stars, not me. Colleagues, collaborators. Ted is my longtime collaborator on just about everything. Collaborators at UT Health San Antonio, funding, of course, NIH foundations that have funded us. And then the Southwest Transplant Alliance, which is this organ donor network, and of course, the organ donors. So, thank you.
You might remember we were talking a little while ago about migraine being a common phenotype to multiple different causes. We particularly would talk about that in the context of Botox only helping a minority of people suffer from migraine. I just wondered if you'd like to expand on that a little bit. So you call migraine a disease, but there's a phenotype. But do you think it's what is the likelihood that there are more than one major mechanism that's driving a common phenotype?
Yeah, glad I left time. This is a fun discussion. So what it is migraine one thing. So there's two major categories. There's migraine with and without aura. I didn't really talk about aura. Aura is a typically visual phenomenon where there's blind spots in the visual field that can start as a pinpoint and grow. There are jagged lines, geometric patterns, colors, flashing lights, they move. That distinguishes people with aura from people who don't get. Most people with migraine do not have it. 70% of the migraine population is not. I think those are two mechanistic different diseases right off the top. But just focusing on the migraine without aura population, which is the majority of them, I don't think that's one. This is 15% of the Earth has migraine. I don't think it's the same mechanism. Well, I think certainly limited ways that the nervous system displays this attack phenotype. It happens to look similar. Or maybe it's just the case that we're actually not good physicians or not good humans are just not good at detecting the subtle differences that exist. And there are potentially many that we just can't see. You and I talked about this when we talked last time. The field technology is getting better at putting devices on people, tracking them over long periods of time. And I think we're going to learn a lot about how people with migraine are different, how their individual attacks are different. how the things that cause their migraines are different, how their daily life is different. And hopefully we'll be able to subcategorize people better, because this is really a limitation of trials. If you have a mechanism, let's say part tube is a mechanism you think is important, but it's only important in a subset of migraine. You got to get those people in the trial, otherwise the trial fails. So how do we identify those people? We have what I think are some convenient ways maybe to do this in two contexts, but for any new therapeutic opportunity, you've got to get the right population of people. And we just don't know who those are. So I talked about this, how does Botox work? I don't know. Nobody knows how Botox works for migraine. It does in a small subset of the population. It's life-changing for them. For most people, it doesn't work. So how do you get the people who are going to respond to Botox in your study? Or if you're a physician, how do you know? I mean, the Botox injection protocol is 31 injections. Look that up. It's 31 injections of Botox in the forehead, the scalp, the head, the neck. That's a non-trivial amount of toxin. The most toxic substance on earth. That's a non-trivial amount of toxin that you're injecting into people, and you know that many of them are not going to respond at all. So how do you avoid it? This is a thing. You know, I don't think this is limited to migraine.
No, it's exactly the same problem treating dementia, even Alzheimer's is the same thing. It's probably
patient stratification.
yeah, it's going to be super important.
Yeah, I mean, fortunately, we have a lot of people. People have a lot of migraines. Most of the people with migraine are otherwise healthy people. There's really nothing that you can find that's wrong with them. They just have migraine. So they're living their lives. They're active. They're doing things, and we just need to get devices on them and monitor them over long periods of time in large numbers. So anyway.
I was curious. So I'm an occupational therapist and kind of like patient outcomes and what kind of people are doing. So my question is like, how, like just to guess, how many people do you think are out there with maybe nerve or pain related issues that might get misdiagnosed as migraine due to kind of just the general nature of what we know? So, you know, maybe they go in, a neurologist is like, I'm not sure exactly the mechanism of how this disease works. Most people have migraine, sixth leading cause of disability, and you probably have migraine. And kind of trying to pin down like these outcomes, because then let's say like you're giving somebody a CGRP or another drug and they're like, it's not working, but we don't know enough about what works based upon what person. So how do you kind of like tease all of that out?
I wish I had a good answer for you, but I don't. Migraine is often diagnosed as an exclusion to other things. That's a very common way to get around it. So certainly if you have aura, if you, the first time you ever have a migraine attack, have blind spots in your vision that are growing, and people often have aura that is aphasic. So they can't speak. They're speaking, the words coming out of their mouth, it just doesn't make any sense. If the first time you ever have a migraine, blind spots, your language is incoherent, you might imagine that you're going to end up in an emergency room very quickly, and you're going to have all kinds of scans done, all kinds of tests. They're not going to find anything wrong with you. Will be diagnosed as probable migraine with an atypical aura. And then when that happens again to you later, now you're sort of ready for it because you've seen it before, and that happens a few times. Now you get the official diagnosis. There's nothing specific. It's just because we can't find anything else wrong with it. So I think this happens a lot. Migraine is comorbid with, just by statistics, it's comorbid with lots of things. Right? It's comorbid with gastrointestinal pain disorders, with low back pain, with other types of pain. It's comorbid with anxiety and depression. Just as a very common comorbid thing. And headache is a symptom of almost anything you can imagine. It's a very non-specific symptom. Migraine is not just a headache, it's a very specific type. Headaches in general are very common, often can be misdiagnosed as symptoms. So I don't have a good, I think this happens a lot. Whether or not people actually get relief for whatever it is their primary disease is by taking migraine therapeutic is a good question, because these drugs have been tested. The migraine-specific drugs have been tested for other conditions and don't seem to work. So it's possible that you think you have migraine, you get prescribed a migraine drug. That's actually not what you have. Drug doesn't work because you don't have migraine, or maybe it just didn't work for your migraine anyway. And now you're in a difficult spot as a patient. Now you're going to go through a number of different kinds of specialists, and people aren't going to know what's wrong with you. This is the problem with the disease that doesn't have an objective thing that you can look at. And a blood test or a brain scan, there's nothing really that you yet that can clearly diagnose you as having migraine. So this is just unfortunate part of that.
Interesting too, because I feel like I like that phrase you said, where it's like, if it's not anything else. Because I know a lot of times when I'm in the clinic, I see people automatically default to migraine. And I always keep thinking, but what if? That's very interesting.
Yeah, certainly want to rule out, strokes and really life-threatening things, but that's usually relatively straightforward out. And then now you're just in the
What about the safety of some of these drugs during pregnancy, since prolactin is such a likely factor?
Yeah, I mean, that's almost certainly going to be a contraindicating. Now, there are a number of drugs on the list that I showed you. You couldn't see it because there's too much text on the list, but there are some anti-epileptic drugs that are known teratogens that have been used preventatively for migraine for decades. And there's very specific counseling that has to be done. Women are prescribed those drugs. They often have to be on birth control. But the majority of people with migraine are women. The majority of them are in reproductive years. And so we have to think about how that population is going to be able to tolerate the drugs that we give. So there's no easy answer for that. I mean, you need safe mechanisms that pregnant women can tolerate. I don't think prolactin is going to be that mechanism. But again, I mean, there are plenty of drugs that we know are not safe that are used. That's a whole separate discussion about whether that actually should, because often, women don't know they're pregnant sometimes. So that's a tricky. Yeah.
First, as chronic migraine sufferer myself, thank you for being one of 10 labs in the country that researches this. As you've talked about, and as Mick highlighted, there's a huge variety in associated symptoms and comorbidities. And I do know some of the associated symptoms are also migraine triggers, so there's interesting effects there. Has there been any effort to try to, beyond differentiating aura versus non-aura migraines, to try to look at, to categorize subtypes of symptoms or comorbidities or triggers that typically hang together to maybe start to handle this issue of actually categorizing the possible causes of what's happening here?
Yeah, it's a great question. I wish there were more of this, and it's somewhat hard. It's often hard for people to remember all of these things when they go see their physician. So a lot of this is anecdotal and just whatever it is they were able to tell. Sometimes it matters what the physician is actually asking about. You don't ask, you don't hear the answer to the question. Migraine diaries are super helpful, but that's a very time-consuming, laborious thing to write down every detail of your life. So this is another area where I think devices, smartwatches, I think are still relatively primitive, and the kinds of things we're putting on the body right now looks like a watch. It won't look like a watch in 20 years. It'll be something more sophisticated. I think that's actually going to help us to make answers, to come up with answers to these kinds of questions, because it makes it easier on the person. They don't really have to worry about tracking. It's sort of tracked itself. Maybe they can input things like, what did you eat today? That kind of information. We've got to make it easier on people to do that. Because I would love to know the answer to, is there any prediction that a physician can do for who's going to respond to what kind of therapeutic, just based on what they're told? Many headache specialists that you talk to will say, yeah, somebody comes in and they say XYZ, I'm going to give them this drug because I've seen that before. That doesn't often end up in a paper. Unless I talk to that physician, I don't know that. That could easily turn into a hypothesis for an animal study. So I think there needs to be way more of this kind of thing than there is right now, and we just need to help people do it, help the physicians track it. So there's good apps now, migraine phone apps. They're interfacing with smartphones, tracking people over long periods of time. Migraine Buddy is I don't have affiliation with them, but it's the more common one. It's 4 or 5 million users in the US that are on it, right? So that's a data source. And so use it. Like, it's helpful to the research community to know about you. But I hope 10 years from now I have a better answer to that question.
So in thinking about some of the sources of variability, the data on epidemiology of this, are there particular, aside from the sex differences, age distribution, are there vulnerable spots, urban versus rural centers, or locations where there's greater exposure to certain environmental toxins? Is there any other hint?
Yeah, there's actually quite a bit of that kind of thing. Environmental factors, contributors, cigarette smoke, gasoline use, bleach, there's all kinds of chemicals. It does seem to maybe be more common in urban areas. Is that always just environmental exposure? Is that to stress? It's hard to know that. There's differences across the earth. Caucasians have the highest percentage of migraines. Africans are the lowest. Asians are sort of in between. It's not really that dramatically different, but it is actually different. So it exists. There are data on these kinds of things, but it's hard to know what they mean. At least yet. There's a socioeconomic difference. The higher up the socioeconomic ladder that you go, the lower the prevalence of migraine is. And is that because people are less stressed? I think so. Is it diet? Can they afford to eat differently? I mean, that's 10 hypotheses that you that.
Yeah, it's found.
So it's an observation. It's some mechanistic work behind it. It's kind of hard.
I think
I'm happy
we'll
to-
have to. I'll go to the. Any questions online?
Yes, we have one question. It goes like this. Could NO donor be forcing an effective impaired synthesis of NO vasodilation that provokes alarm signals to sensory neurons that produce pain?
So great mechanistic question. The link between nitric oxide and migraine is not clear. We don't. I have, depending on how far I got in this talk, I had another set of slides. You might have, what do blood vessels have to do with migraine? This is a whole interesting story in and of itself. Nitric oxide is a vasodilator. CGRP is a vasodilator. Any number of other kinds of substances that are vasodilators trigger migraine. So why does that cause a migraine attack? Triptans are vasoconstrictors. Caffeine is therapeutic for many people with headaches. That's a vasoconstrictor. So what is this vascular contribution to migraine? We've got preclinical work on this. There's some clinical work on this. I think there's a connection between the vasculature and migraine. We don't understand it. Many people in the field have tried to write this off because if you have someone in a scanner during a migraine attack and you load them with a contrast agent, you look at their vessels, they're not actually dilated. So, that just maybe means that dilation is not the problem, but that doesn't mean the blood vessel isn't the problem, because blood vessels can signal to neurons and vice versa in many different ways that are not dilation. So I think that the nitric oxide phenomenon is exposing some vascular contribution to the disease that we just don't yet understand. The issue, because I think you mentioned endothelial nitric oxide synthase in there, we know that inhibitors of NOS are therapeutic for migraine. So we know that. That's been shown in a small trial. Problem is it elevates blood pressure. FDA is not going to let us do that in humans, so it's never going to get approved. So we have to figure out what the mechanism is and get around the vascular blood pressure issue. But I think there's absolutely something there, and we just don't know what it is yet.
And on that somewhat optimistic note, I'm going to just thank you so much. I think it was interesting for all of us and really a great way to think about how complex issue can be very well scientifically approached. So thank you very much.
Thank you.